And she has been working on the chronic pain and neurodegeneration, and also their group has been working on the small molecules for ion channels.
Okay. Good morning, everyone. I would like to start by thanking the ICMS Organizing Committee for the opportunity to present my work on ion channels here today. It's a pleasure to be in this meeting. So today I'm going to talk about ion channels at the nexus of the pathophysiology and the treatment of neurodegeneration. So the type of neurodegenerative disease I'm interested in is motor neuron disease. This disease is highly prevalent in Australia as well, North America and Europe. It is a disease that is characterized by the neurodegeneration of motor neurons on the cortex as well as lower motor neurons that innervate the muscles. We do have a high prevalence in older individuals, being males more highly affected, and we do have a few cases of neonatal neurodegeneration. There are 4 types of motor neuron disease, and they are separated by the type of the neurons they affect. Amyotrophic lateral sclerosis is the most prevalent form of motor neuron disease, and it affects both upper motor neurons in the cortex as well as lower motor neurons that innervate the muscles. And the median survival is 2 to 5 years after disease onset and diagnosis. We also have all the 3 types, and they are separated by neurodegeneration of only lower motor neurons or upper motor neurons. And I'm not going to talk much about these 3. I'm going to focus today on the ALS. So ALS, amyotrophic lateral sclerosis, is classified in 2 types. We have familial, which are well characterized by genetic mutations that lead to neurodegeneration. But we also have the sporadic, and the sporadic, they are the majority, they're 95%, and the causes are very unclear. The familial ALS causes, we do have mutations in the SOD1 gene that supermutase oxidase dismutase, superoxidase oxidase dismutase 1, which is an enzyme that controls oxidation in the membrane, in the mitochondria, sorry. And we also have some other mutations like C9orf and TDP43. And today I'm going to be focusing this particular mutation here, and because it is the mutation that is highly prevalent in the sporadic individuals that will acquire that later in life as a somatic mutation. So the pathophysiology and treatment for ALS. So as I mentioned previously, the pathophysiological mechanisms are very complex, and the disease cause or causes are completely unknown for the 95% of the ALS patients. But they all have one pathophysiology in common, which is the motor cortex hyperexcitability is imperative prior disease onset. And this has been identified in both humans as well as in preclinical models of ALS. So I'm going to be looking into this hyperexcitability in the motor cortex as our main pathophysiological characteristic for studying. There are very limited treatments available for ALS. Riluzole, it's a non-selective ion channel inhibitor. It is the only FDA-approved drug for ALS that actually can modulate hyperexcitability. Unfortunately, it does not help much in terms of prolonging the life. ALS can be very variable. among the individuals suffering from ALS. Sometimes we have their life being prolonged by 1 to 2 years, or sometimes we have cases like Stephen Hawking that could live 10 or 15 years with the disease. And they postulate that one of the main mechanisms of action of riluzole is actually blocking sodium channels. So here's just a representation of the synaptic crosstalk, the presynaptic end of the neuron. We have the sodium channel here, which is a key mediator of neurotransmitter release that will be activating the next neuron. And they also know that glutamate exhausts a high level of neurotransmission mediated by glutamate is present in this toxicity. So that's the mechanism they postulate for riluzole. But riluzole is a small molecule and it's a very promiscuous drug. And if you look at the literature, they target not only sodium channels, but a range of other ion channels. And we believe that this can be one of the limitations of the mode of the therapeutic effect. Beyond that, we have edaravone, which is an antioxidant that helps the individuals settling their symptoms, but it does not prolong survival. So treatments are very non-existent. They're pretty much non-existent for this type of disease. So in my lab, we are very interested in understand how ion channels contribute to the pathophysiology of neurodegenerative diseases. So with a collaborator, also at the University of Queensland, we start to study motor neurons which are derived from humans suffering from ALS. So I'm going to show here today data from this particular individual. It's a female, 70 years old, where she donated her fibroblasts, and we can with her cells, we can reprogram them in iPSCs, and then program them in neurons, and later on in motor neurons. So I'm going to be talking about assays that we developed with this type of motor neurons here. I'm going to touch base quickly on that one. And for the familial individual we have is a male 50 years old. Unfortunately, we could not get the protocol working for this particular individual, and we are still trying to understand why it's such a different, although it's the same disease, the causes here are very different for what we are seeing here. So it's very heterogeneous, which means that a more personalized drug development approach should be used for this type of disease. So we did start to probe these ion channels on these motor neurons derived from ALS individuals, and we used initially an approach where we measure intracellular calcium signaling and using this platform called Flipper. And at first glance, what we saw was that healthy individuals, which are the same sex and age matching, they would respond nicely. For example, here the green traces where we have addition of a pharmacological agent that will open sodium channels, for example, and they open and close nicely as expected. But with the ALS individual, we see the first phase, it's non-existent, and actually it develops continuous repetitive spiking of this calcium intracellular signaling. And more interestingly, when we add product of the mutation, that is the most common mutation found in individuals with sporadic ALS, which is derived from the C9orf extension. It is a dipeptide, which is a repeat of proline and arginine. We see that this activity becomes enhanced in the ALS individual, as we can see here in blue. But for healthy individuals, they are very insensitive to that. So we were quite surprised to see these results. And we really start to focus on what is actually happening here with these individuals with ALS and why we have this repetitive spiking happening. So we thought that that would be definitely a good indication of that aberrant hyperexcitability that we see in the motor cortex. So we went and we did a deeper analysis of how this PR20 molecule. It's acting on the ALS individuals, measuring rise of action, decay of action, intervals, duration, and amplitude of this spiking. And what we learned initially was that, for example, when we stimulate sodium channels are highly expressed in motor neurons. We can see that they do have increased rise in action, but when we add the molecule PR20, and this is a molecule it's added at low nanomolar concentration, here I think was around 50 to 100 nanomolar, we add to our preparation, it really increases the spiking, the rise of action, it becomes longer and slower. And when we activate the same cells with activator of NaV1.6 channel, and we are interested in the NaV1.6 because it's one of the sodium channel subtypes that is mostly expressed in motor neurons. And where we see that the NaV1.6 is definitely altered in the presence of the pharmacological agent that we added. And interestingly, when we add the PR20, there was not much additional effect in this case. So this is something that we are looking very carefully now. When we stimulate calcium channels, for example, we see that calcium channels by itself, they will not actually have any dysfunctional calcium signaling, but when we add the PR20, then the calcium channels start to become a problem as well. And we also look at the glutamate receptors, which in fact, we did not see much change. Actually, there was a rapid increase and a rapid decrease of this calcium spiking. So, and this, we look at other parameters as well, and I just want to highlight here how there's NaV1.6 in the presence of PR20, it does not produce significant, or sometimes we have additional effect here and also in the duration, and sometimes there is no additional effect here on, for example, action potential intervals. So this information was, for us, it feels that it was quite difficult to make interpretation at that moment we collect this data. So when we start to look at more carefully how we were activating these ion channels, we noticed that the NaV1.6 activator we use in this case, that is a scorpiotoxin, it actually has very particular properties. And then we start to realize that what the binding site of this toxin, it's probably very similar to what the PR20 is doing to the ion channels of the ALS individuals. So we are focused on that to see if we understand a little bit more of what's happening with these ion channels in ALS. So here, just to highlight again these traces. So OD1, that's the scorpion toxin, which is a selective toxin for NaV1.6. In the presence of PR20, it does not look the blue here, OD1, and you have the red here. We don't see much difference in the traces. So we, as a pharmacologist, I see that looks like OD1 and PR20, they are actually competing for the same binding site, which is a quite interesting information for us to understand how PR20 is actually acting on the sodium channels of ALS individuals. And we have the veratridine here, which is a sodium channel activator that actually binds to a different site. So, and that's this pink line, that's the calcium reading we have the veratridine, but when we add the PR20, suddenly the signal becomes very similar to the OD1. So we are really starting to realize that might be that OD1 is targeting a site, which is the same site that the PR20 is acting to cause dysfunction in the ALS individuals. And so then, as a Venon's researcher, we start to actually dig in more details of how this could be happening in the ALS individuals. And it's important to highlight that health individuals, they are insensitive to PR20. So it's something that it's part of the pathophysiology that happens only in the ALS individuals. So we moved to actually explore site 3 on the sodium channel, that's where OD1 binds, but as well as site 4 as a therapeutic approach to actually stop the repetitive calcium spiking induced by the scorpion toxin in ALS. So just representing a little bit in more detail, so we have OD1 that targets site 3 of the sodium channel to open the channel, and we have here a peptide that I'm calling Propatex, which tags the site 4 to block the channel. And here's a typical electrophysiology trace. Peptides that target site 3, they will delay fasting activation, and peptides that target site 4, they will inhibit the peak current. So we then moved to make some tests with the ProPTEX in the actual ALS-derived motor neurons, still using the calcium imaging. And we also test, in this case, riluzole for comparison. Riluzole is the FDA-approved drug for ALS, and what we learned was ProPTEX was quite more potent to modulate this repetitive calcium spiking at low nanomolar concentration. But in the presence of PR20, the cells became more resistant, about 10-fold. So there is an influence there. And when we test the riluzole, we had the nanomolar, 422 nanomolar, showing that ProPTEX is way more potent than riluzole. And again, in the presence of PR20, the cells become more resistant to this inhibition. And just to highlight quickly here, a different type of ALS we had accessed the cells for. It's the ALS with front temporal dementia, which is quite common as well. And in this case, we noticed that the presence of PR20 does not alter the sensitivity of the cells, which was quite interesting. So we are talking about here probably a different type of ALS, which is not driven by the PR20. So this is just to show you how heterogeneous the disease is. So with this data in hand, we start to look into how ProPTEX could actually modulate aberrant hyperexcitability in SOD1 mutant mice, which is the only model which has clinical relevance for ALS. So doing manual patch clamp, we patch motor neurons in the layer 5 of the motor cortex and measure the actual potentials. And we learned that wild-type animals control, they will start to fire when we inject around 200 picoamp of currents, but the SOD1 animals, you look at control here, they will start to fire way earlier. So this is a characteristic of these channels are being hyperexcitable. And when we treat these brain slices with ProPTEX 50 nanomolar here, we can see there is a reduction of this spiking here, and then there was actually some reversal of the spiking after 20 minutes washout. And with the SOD1 mice, we noticed that the 50 nanomolar of ProPTEX actually can bring the firing back to normal here, looking like the control here. And in this case, what was quite surprising, we could not wash out this activity on the SOD1 mice. So the sodium channels, particularly the NAV channels, on this ALS, they are definitely altered compared to the wild types. So then we did a full characterization of ProPTEX on upper motor neurons, and we learned that it has a low 50 nanomolar, around 50 nanomolar IC50 to modulate that, as well as in lower motor neurons, which were a bit more sensitive. So here we did motor cortex as well as hypoglossal. And hypoglossal, it's a nerve that innervates the tongue. And when individuals go to the doctor for the first diagnosis, it's usually when they start to have failure on the speech. So that's quite an important nerve that we are looking at, a lower motor neuron. And we see that ProPTEX actually can modulate both upper and lower motor neurons in wild type as well as in SOD mice. So, and this did not make much difference here, which that's always a concern for us if the actual, what we develop, it's actually targeting the disease dysfunctional channel. And it's important to mention as well that we did try to test all the spider peptides as well, and only ProPTEX had a very clean inhibitory response that we could actually follow up to characterize. So we postulate the main targets for ProPTEX to be causing this modulation are NaV1.2 and NaV1.6. I don't have time to explain the whole hypothesis around it, but you see that these are the channels that are mostly highly expressed in the motor neurons, and they are very involved in neurotransmission. And we do have an important off-target, which is the NaV1.4, where we we want to avoid this target, and every optimization we do, we make sure that NAV1.4 is not being targeted. So we did produce, made lots of rounds of optimization trying to improve affinity for NAV1.2 and NAV1.6, produce them chemically, and here we have a profile, and this was done with the Q-patch on Sophion, where we could analyze lots of leads as well as lots of ion channel subtypes. And I want to highlight here two particularly optimized peptides, OPT4, where it has higher affinity for NAV1.2 and lost affinity for NAV1.6, and OPT6 that we gain potency for both NAV1.2 and NAV1.6. So then we moved to test this on zebrafish model, and we were quite excited about it. This is a model where there is a motor neuron degeneration, which is gradual, progressive, and we know the sodium channels in the zebrafish, they have the same binding site as the human channel where these toxins bind. So we thought that that would be a good model for us to start to test our candidates. And what we learned is Propitex, it really helps to recover swim behavior on these animals. So we have a control normal zebrafish. Then we have a zebrafish here we call SMA, which lost, there is the progressive neurodegeneration of their motor neurons, and they lost the ability of swimming. And here we have control with riluzole, the FDA-approved drug, and here we have Propitex that was able to recover, to protect the neurodegeneration, and the fish continue to swim normally. And then when we look at survival, then we found a more striking result. While the Propitex, it really increases by 22% the survival of these animals. When we remove the activity of NAV1.6 and we push the pharmacology towards NAV1.2, we actually lost the protective effect. And then when we test another peptide where we had enhanced the affinity for both NAV1.2 and NAV1.6, we actually improve the survival. So here I think we are really seeing what are the actual targets of these therapeutic effects, which here we postulate they are NAV1.2 and NAV1.6, particularly NAV1.6. So then, finally, we moved to do more complex in vivo models, and we test Propitex, and we are in the process of testing all the optimized peptides on the SOD1 model. So for this administration, we do have intracerebral ventricular cannula implantation. Unfortunately, it's a very invasive procedure, but to avoid any potential systematic effects that will be hiding the therapeutic effects we're looking for, we opted to this procedure now, and these animals then go to motor function evaluation. And that's quickly the design of the experiment. So animals with 50 days, they receive the ICV cannula implantation. 1 week after, they start to receive treatment. The treatment is twice a week. And from P70, we know this model start to present the disease, motor function defects, and here we are measuring this behavior. So we have very encouraging initial results. So these results are from P100, where we have here wild-type control, the time to fall. So this is a beam that keeps rolling. So we measure the time the animal takes to fall, and then they fall. That's our measurement. So we have here control as expected, take around 150 seconds until they fall. And then we have the SOD1 mice. They are very debilitated, so they don't last long in that beam, showing that there is muscle weakness and loss of control. And then here we have wild type treated with our peptide, and we were quite happy to see this result. Looks like there is no side effects that could be concerning. And more importantly, the SOD1 mice, when they were treated with our peptide, they really protect the motor function. So these are very interesting results we captured just a few months ago, and now we are in the process to validate this further to understand how NAV1.2 and NAV1.6 are participating in these therapeutic effects. So I would like to thank our team, the Cardoso lab, particularly Jessica, Charan, Refik, and Van Tran that produced all the data I showed here today. Our collaborators, Mark Bellingham, he was the first one that tested ProPatex in motor neurons in his lab, so he actually really had an important role in triggering this project. Shu Nigel, he does the IPS cells, the motor neurons we test, in Jad Giacomoto that does the zebrafish, and my mentors from the University of Queensland, Richard Lewis and Glenn King. Obviously, people living with ALS, they donate their cells to us, and these people, they are really hopeful that we can actually come up with a solution for them. Unfortunately, the reality of science is not that simple, but I would like to really emphasize my appreciation for patients that donate the samples to us. The venomous organisms for developing these amazing molecules, they are highly selective, and we know their mechanism of action. Sophion for the invitation, and also the funding agents, the US DoD from US. Thank you.
Thank you very much for a wonderful talk, Fernando. Please, yeah. Yeah, you can make.
Thank you. Lovely work, as always, Fernanda. Your last data slide on the rotor rod tests, that far right panel with the SOD1 Propotex compound, do you almost think that's two populations that are sort of merging into one streak of...
Yeah, it's not very homogeneous, but what we have here as well, this is a very complex experiment, and there is difference in male and females, which I'm not showing here today. So the males, they have a more drastic disease, which I believe is this group here. So they do have recovery, and we actually have new measurements on this stage that this becomes clear, the therapeutic effect on the males, but these are mostly females. So for some reason, there is a sex difference, and that's why now we are repeating the experiment with a larger number to get these numbers cleared. Yeah, but there are differences, and I think this is what we see here.
So it's almost possible that you may be getting that male-female difference whereby the females recover far better, or, they don't have an originally, as poor starting point with the SOD1 mutation. And so, yeah, maybe with more studies, you'll almost get the two subpopulations really splitting, where you've got almost, limited recovery, and then you've got actually a pretty good group that recover very, very well. And it might be sex dependent, it might be other factors that could be causing that sort of...
I believe sex dependent, it's one major issue, but there is also another measurement that we are doing right now, which is the survival, because we are here now at the end stage, and these mice are starting to unfortunately die of the disease. And we know our survival is also increased, but we have to wait for the end of the experiment to release the data. So, and I can tell you in advance that even the model itself, without treatment, there are male and female differences. My electrophysiologist says that our PMS, our hormones, are helping us. Okay. That's the best explanation I have so far.
Thank you.
Please make a short question.
So in terms of the mechanism, I suppose the SOD mutations are loss-of-function mutations, and they may stabilize the NAV expression in the plasma membrane. Is that the mechanism? Have you looked by something like that, immune fluorescence or measurements by electrophysiology?
We are trying a lot to reproduce what we see in the ALS in cells that are not ALS affected. And that would be our first question. So we are trying to stress the mitochondria in hope that will alter the sodium channel activity, but we cannot see that in healthy cells. Only stressing the mitochondria does not give alteration in the sodium channel activity. So we're looking at stressing the reticulum and the plasmic reticulum as well, and try to create an environment that might resemble ALS in hope that we can reproduce this sodium channel dysfunction, but so far we have failed, and we can see that happening only with the cells from the patients.
I mean, I think it's known that superoxide is degrading or is at least removing voltage-gated sodium channels from the plasma membrane, right? So could it be that if you have mutations in SOT that you actually you stabilize more of the protein in the plasma membrane? That was my question. Right? And could that be a common mechanism for the other familial mutations too, right? That you just have more of the protein in the plasma.
Yeah, a higher expression of
Yeah.
the channel, but this is very controversial as well because there are hyperexcitability and hypoexcitability as well. And when we try to measure that expression level of the sodium channels, it does not correlate directly. And that's something that we want to measure the sodium channels expression, these ALS cells. It's to be done, but we don't believe it's a very accurate, there is not a very accurate correlation. And that's why we are not very trustworthy on that hypothesis just yet.
Okay, thank you. Time is out. Please join me in thanking Dr. Cardoso. Thank you. So next speaker is